All right, finally, we're going to talk about phylogenies, and phylogenies are a result of evolutionary forces acting for a long period of time. And eventually, evolutionary forces act to separate populations into different species. So we saw this, and we didn't see this, but you would cover this in an evolutionary class, for example, on Darwin's finches that occurred in the Galapagos Island, is that you started with populations, you started by populations of the same species, and then due to the separation, due to the actions of natural selection, due to the actions of genetic drift, eventually those populations are no longer able to mate with each other and they become independent species. And so what a phylogeny allows you to do, it allows you to use genetic differences among present-day species, and it is used to reconstruct their evolutionary histories, and that's what referred to as a phylogeny. And a phylogenetic tree represents lineages over time. So branches come together at points when speciation has occurred. And monophyletic groups consist of an ancestral species and then all of its descendants. So this is an example of a phylogeny. And so the idea behind a phylogeny is that you are reading backwards in time. So present day is shown here. And so at present day, there's 3 different species that we're looking at in this phylogeny. And these species can be whatever you want. So maybe it's, let me think of some good examples. So maybe it is rats, mice, and house mice. So the idea is now there are species, field mice and house mice, that can't breed together. They're independent species. Rats and field mice are independent species. If you go back in time, eventually you will reach the point where the field mice and the house mice originated from this same ancestral species. So we're going back 10,000 years, 100,000 years, 1,000,000 years, 2,000,000 years, and it depends on the exact species, but eventually you get to the point where they actually were the same species. So there was a point in time where field mice and house mice did not exist as separate species. So this is a node. So this indicates where a speciation event has occurred and one species has become 2 different species. So this preceding here is whatever the ancestral mouse species might have been. And then this ancestral mouse species diverged into 2 different species. And then you can also do the same thing with rats, for example. And if you go back far enough in time, again, there is a time in our history where there were not rats and mice, but there was some sort of species that was a combination of both. And so this phylogeny represents the time where that divergence occurred, where one species became 2 species, one of those species became rats, and then one of those species eventually grew to become different mice species. So this is the idea of a phylogeny, and the important thing to note is that the branch length, this is a branch, indicates how long ago in time 2 species were the same. And so you used existing genetic variation between species in order to infer this information. So if we looked at a gene like hemoglobin, we would note that there's hemoglobin. We would note that there are differences in the amino acid sequence between the rat hemoglobin, the field mice hemoglobin, and the house mice hemoglobin. But the number of differences are dependent by how long they have been separated from each other. And so this is the idea of a molecular clock. So it allows us to look at nucleotide changes in genes that are conserved in order to predict the divergence time. So this is just looking at divergence time based upon fossil records. So this is an independent way to try to estimate when species diverge, looking at bones, leftovers of bones, and then also looking at the sequence divergence. So if you look at how different all of the genes are. And so you see this very linear relationship, this nice linear relationship in the number of synonymous nucleotide substitutions, the amino acid substitutions, and then the non-synonymous substitutions. And so what that means is that there's a clock. The longer things are diverged from each other, the more amino acid differences there are. So this is a very powerful way to just use DNA information. So you can just use information obtained from the sequence of DNA in order to predict how long ago two species have diverged from each other. We're also using that in order to study human evolution. So there's been a lot of really cool papers that have been published in the last 5 years that have occurred because researchers have figured out how to isolate DNA from bone fragments. So we have all of these existing populations of humans, so African, French, Han, Melanesian human populations, and there are also bone fossil remnants of human populations that have existed in the past. So Neanderthal and Denisovan populations, these are fossils and bone fragments, actually bone fragments, I shouldn't say fossils, they're bone fragments that have been isolated from caves, or they have been frozen in icebergs, in snow, in permafrost, and then identified by researchers. So these populations no longer exist. There's no longer Neanderthals or Denisovans that live today, but there are remnants, there are actual biological samples that we now are able to isolate their DNA from. And so what human genetics researchers have found, is that modern day humans actually have Neanderthal and Denisovan DNA within their bodies to a different extent depending on which population you belong to. And so this indicates that there is actually interbreeding between humans and Neanderthals and humans and Denisovans in different parts of the world. So this has changed our view of modern day human origin, in that humans have left Africa in multiple waves. So initially 350,000 years ago, Neanderthal and Denisovan ancestors left Africa and migrated throughout Eurasia. And then about 60 to 70,000 years ago, some human ancestors left Africa, and then kind of waves of humans left over time. So they have also left at 50,000 to 55,000 and 45,000 to 50,000. And these humans have kind of taken different pathways through the world. And some of these humans have encountered Neanderthal populations or Denisovan populations and clearly have interbred with those populations. So, a small amount of this Neanderthal DNA remains, indicating that, maybe 4% of matings back in time occurred between these humans that came out of Africa at a later time period, and then the Neanderthals that had been living for hundreds of thousands of years in Eurasia. And kind of the purpose of this is still under debate. So was this natural selection so the Neanderthal DNA that sticks around in human populations, has that been selected for? So one possibility is that since Neanderthals had been living in these areas for a long period of time, they had adapted to certain viruses and pathogens that lived within these regions. And so humans were able to breed with Neanderthals, and then they were able to obtain these resistance alleles as a way to survive against pathogens. Or it could just be genetic drift. We really don't have a huge understanding of that. But there's researchers, including people at Georgia Tech, Joe LaChance, who are studying these problems right now. So that is the extent of Chapter 26 on population genetics.